Okay, in the two previous videos we talked about the microfluidic reactors, which can be used for the precise control over the size of the nanoparticles which are synthesized, and we also talked about measuring the diffusion behavior of the three-dimensional structures of the cells, different layers, and we can detach them to analyze the concentration profile in each of them. In this video we are going to talk about the biosensors. For example, consider this primary tumor that we have and the blood. You know that the amount of cancer cells which can be found in the blood is reasonably smaller, lower compared to the blood cells. So it won't be possible to have a quantitative measurement of the concentration of the tumor cells with the normal blood tests. So these tumor cells are not going to be recognizable. You cannot detect them. They are not detectable. So in order to detect these, you can use these microfluidic chips which have the microposts. These microposts have a sort of antibody which is which has the affinity towards these tumor cells. So when the tumor cells are passing, they will stick to these microposts, and after that you can measure the amount of the cancer cells which have been stuck to these microposts to have a quantitative measurement of the amount of tumor cells which was previously in the blood which flew here. Well, we covered this. Let's have a look at this slide. This is also about the sensors that we can use by the nano devices. You see here that we have the single-stranded RNAs, and if we have the sensor here and there are other single strands here, if the strands present on the sensor are complementary compared to the strands which are connected to these gold nanoparticles, they will self-assemble here. When they self-assemble, the concentration of the gold nanoparticles here increases. For example, by measuring the conductivity of the solution within this process, by measuring the so-called impedance. Impedance is just the resistance. Okay, impedance is just nothing but the resistance of the solution through the electronics. You can measure, for example, how many of these have been adsorbed here. So by the change of the impedance, you can have a quantitative measurement of the amount of these single strands here that you had before, which are complementary towards the single strands on the surface. Well, we can also let's say measure the change which happens to the amount of, let's say, fluorescence signal that we get from these molecules. For example, if they are the fluorophores, when they are closer to each other, some changes will happen into the amount of signal that they can say, and this again gives us a quantitative measurement of the amount of let's say single strands that we had before here. So you see that the changes in the impedance and the signals can be used to create the sensors. Well, here is another example that a magnetic field has been applied so that the gold nanoparticles form the nanowires, and they can be used for the redox reaction. So again, the impedance is gonna be changed so that You can use it as a sensor. Again, here we have the sensor here, and depending on the amount of the drug molecules which are adhered here, you see that the impedance is gonna change. For example, when there are a lot of drug molecules, the resistance of the solution increases, so that the passage of the electrons will decrease, so the impedance has been increased. So again, it can be used as a sensor. Here we are gonna talk about the nanowires, which can be prepared by different layers of materials. For example, we have these templates, the first material, then we add the second material, then we add the third material, and then we will add something, for example, like an acid or a specific type of salt, to degrade it, and we will have finally the nanowires which are ready to be used. If we let's say accommodate the central part of these nanowires with the antibodies, and we use them here, you see here that depending on the amount of um, the, let's say, for example, the molecule that we wanted to measure in the solution, depending on the amount of them which are adhere on this surface, the conductance will change. For example, we have the conductance here, and when they are adhered, the conductance will decrease. It means that the resistance, the impedance, has been increased. And when these are detached, you see that we have again the increase in the conductance or the decrease in the impedance. Well, this was all about the biosensors, and in the final part we are going to cover the process in which we heal the wounds.